So everything that Bill said was mostly, well, largely true apart from one important thing, which is that I don't actually know very much about e-science, and even less about e-research. So that's the bad news. So I'm not going to talk about them. Apologies. And it's a little bit of a surprise to me why I was asked to give a keynote at this event. And I'm sure after this talk you will have arrived at the same conclusion. But anyway, I'm here, and I did say yes. I didn't say yes to a keynote, but I discovered I was giving it. So I don't know anything about e-research or e-science. There is an e-science bit of Microsoft, which we have something to do with, and it makes for an interesting discussion sometimes with some of that group. But what I am going to talk about is, I suppose, roundabout where the work of some of the groups that I lead in Cambridge. I'm told, by the way, I've probably got the only group in, well, I have got the only group in Microsoft that's not in Microsoft Research, and in Microsoft that's not computer scientists. So I'm told that it's a nice party piece when for those in my groups, when you say you work at Microsoft and you say, what do you do? And you say, I'm a theoretical ecologist. It's a bit of a showstopper, I think. I think people are expecting you to be a sales and marketing person or selling Windows or computer scientist. But I do have some interesting groups full of theoretical ecologists, zoologists, mathematical biologists, physical oceanographers, chemists, neuroscientists, developmental biologists, and the likes. So all this talk is kind of, I suppose, is a sort of a broad motivation for everything that we do. And I suppose the link to sort of e-research and e-sciences is, I suppose, that I'm going to give you a very personal view of what I think are the most important challenges that we face this century, probably of any century, because this may be the only century we've got to actually do something about them. And what some of the scientific challenges are in order to help address those important societal challenges. And I think some of the scientific challenges are pretty difficult to solve because they represent currently largely intractable problems. And it's not for me to judge. It's maybe for you guys to sort of think more about to what extent e-researchers and e-scientists can make a contribution to addressing them, even if you agree with them. So. Excuse is over. I'll start, I suppose. And I was going. This is kind of an okay title. I was going to call it a codification of heaven, some lessons, but that seemed a little bit too silly, so I didn't. But one thing I do want to start with. I don't know if anybody reads Wired anymore. I could deny that I read it, but it wouldn't make a lot of point. So anyway, in the June version, the main feature was titled "The End of Theory," and its main claim was that e-science is about to, or started to, generate enough petabytes of data to make theory redundant and to make the scientific method obsolete, which isn't a view I really share, but it seems to be one that I hear a lot from people working in e-science. So what they basically this is a quote from the Wired thing in June, which it says, our ability to collect petabytes of data is enabling us to find answers to fundamental questions, making the scientific method obsolete. In the age of big data, more isn't just more is different, which to me more is just more. I don't know what is different about it. But anyway, that's what they said. So I suppose one of the things that goes to my talk, at least, is that, just sort of paraphrase Oscar Wilde, the death of theory is probably, or reports of the death of theory is greatly exaggerated. But I do agree that there's a transformation needed in the scientific method for reasons that I'll come to. So I don't know how related that is to the e-science sort of view of the world, but it's not one that I particularly share. So the first challenge I want to talk about is I think the main challenge in the broadest sense is of understanding complex natural systems from biochemistry up to the biosphere, and why it's a surprise in some sense that that's still a big challenge, not least because we've had this apparent 50 years of spectacular success of molecular biology, and the poster child, of course, was the Human Genome Project. For the culmination of that achievement, which was, and I suppose it's in some sense a poster child for e-science, because it involved a lot of data, certainly, and a lot of computers. And no one would deny that molecular biology has made some remarkable advances in our understanding at a molecular level of bits of life. The problem is, after that 50 years, and we still don't know how living things work, we don't know how, I mean, if it's near the end of 2008, and we have no idea how a cell works, a simple eukaryotic cell. We don't know much about how complex of cells in a system work together to do something. So we don't know anything about, so true to say, we don't know anything about the immune system. We don't know how the immune system functions. We don't know how even a little nematode worm, C. elegans, is this little worm here, who Sidney Brenner calls, quite rightly, I think, nature's gift to science. But we know every single thing about all 595, depending on whether it's hermaphrodite or not, cells. But we don't know how they all work together to produce this chap. Come in, you haven't missed anything, and nor are you likely to. Even though its genome has been fully mapped, of course we know remarkably little about how brains work, and we know next to nothing about how these things on the end work, which is a bit of a shame, really, just to have to admit that. I mean, other than taking things out, sewing things off, stitching things back up, removing things, we don't really know how to fix them when they're broken at a system level. Other than really the mechanical bits. As a consequence, predictive biology is almost unheard of. Preventative medicine, which remains largely a dream, despite the optimistic view of our ability to do this by a friend of mine, Lee Hood, at the Institute of Systems Biology, who talks about P4, which is, I think, preventative, participatory, personalized, and predictive medicine, which is a nice idea. Interestingly, if you think about living things, this is the subject of a talk on its own, but I'll just mention it. We're unable to predict and prevent a global pandemic. Everyone talks about the need to treat all these diseases that kill 2, 3, 5 million a year. When the last global pandemic occurred in 1918, everyone thought it killed 50 million people, Spanish flu, and it turns out now it probably killed about 100 million people, and that's before anyone had invented EasyJet. If you look at even the crude models that exist today of a global pandemic outbreak, if you include the way in which people travel, estimates range from 500 million to 1.2 billion people that it would kill fairly quickly, because of the way in which people, largely because of the way in which people travel. And that would be I suspect that would be only the start of our problems, because there wouldn't be many Tesco's open at that point, and society would break down pretty quickly. And most people are agreed that it's not if a pandemic occurs, it's when it occurs. And this, I continue to find it absolutely remarkable that so little attention is paid to this. We have a project in this area working with actually an Oxford's chap, Neil Ferguson, now Imperial, on building the world's first global pandemic modelling system. I'm not going to talk about it today because it's too depressing. So what we do have, as I mentioned from molecular biology, is we have a lot of facts. We know nearly everything about most of the parts. We know increasingly quite a lot about the reactions between some of those parts. But the problem is that function and behavior of living things is not obvious from the parts, all the reactions. And one of the reasons is because living things are not characterized by parts and their reactions, they're characterized by processes. So simple processes that range from cell division, cell cycle control, immune system processes, but it's an important term that I'll come back to later. But it's fundamentally different from reactions, is a notion of processes, and it's an important one. Anyway, so after this 50 years of molecular biology of taking everything apart, everyone's now decided they have to spend the next 50 years putting it back together. So there's this term called systems biology, which would be nice if it were about systems, or indeed largely about biology. So the systems biology falls into basically a few approaches. So approach one is, which might appeal to the e-scientists in the room, which is collect yet more data and do more analysis and infer something, hopefully, more useful than we've currently got, but from that data. So this is the omics, metabolomics, genomics, proteomics. The list is a growing one. And it does indeed generate an awful lot of data. Approach two is to build a static model of what's a very dynamic reactive system. And this is largely reaction graphs. So this is driven by the work, I think, probably most importantly, of Hiraka Kitano, who works at Sony, oddly. And I'll show you one such graph. So this is a graph of a MAP cascade, which is a mitogen-activated protein kinase. It's a pretty simple pathway, despite the fact that it doesn't actually look simple. And actually what this reaction graph enables you to do is it describes very, very well this MAP cascade. That's good news. The bad news is it's completely unable to explain how it works. So it's a nice description of one snapshot in MAP cascade, but that's all it is because it's static. It just tells you lots of bits about what these chemicals do at one point. And they're not particularly biology friendly, biologists friendly, as you can see. In fact, they're not friendly to anyone. So approach three is to develop continuous models of a discrete system. This is the mathematical biologists. I hope there's no biologists in the room. We'll never get out of here alive. So this is the order differential. The tool of choice for mathematical biologists is order differential equations, which are a great tool, but it's basically give you a continuous explanation of a discrete system, which is limiting, to say the least. And in any of those approaches, we just get more facts. And I think the central problem I have with what many in e-science claim about all this data stuff is that facts are not science no more than the dictionary is literature. And that's a pretty important point, I think, to take away from the talk. I think it pervades everything that we do in my lab. I think the best way to sort of summarize the situation that we're in biology, systems biology, molecular biology, experimental biology is a very quick story about could a biologist fix a Tamagotchi? And I have to apologize to a colleague of mine, Luca Cardelli, who had this idea originally, but I've really ruined it. So we've got our organism, Tamagotchi nipponesis. Anybody can remember these? There were billions of them made by Bandai in the 90s. Every Japanese schoolgirl had one, and eventually everyone had one, and then they all disappeared. I don't know what happened. They became extinct. So the main point I'm making is established scientific approaches to studying complex things, in this case Tamagotchi, all fail. So mechanistic approaches fail. This is the typical well-established reductionist approach to biology. So if you remove the components from a Tamagotchi, that's either ineffective or inconclusive or fatal. A lot of money is being spent on protein folding, and a lot of big blue computers are being switched on and all the rest of it. It's equivalent of this Tamagotchi folding problem, which is a sequence of manufacturing stages in a factory we've never seen, and is all operating in a language we don't understand. It's too hard anyway, and even if you could solve it, it would reveal little about function. So there's not a lot of point in doing it. Behavioral approaches fail. So subjecting a Tamagotchi to extreme conditions, first of all, voids warranty, so don't do it, and reveals little about normal function. And Tamagotchi don't respond to stimuli consistently, and one of the reasons is because there's wide stochastic variation in each one. Ecological approaches fail to understanding Tamagotchi. They're difficult to study in their native environment, which is a Japanese schoolgirl's hands, and of course they evolved in this absolutely unfathomable Japanese consumer goods market, so we don't understand much about their evolutionary path. Mathematical approaches completely fail. I defy anybody to tell me what order differential equations a Tamagotchi obeys, or a sea urchin, or a zebra, or whatever, name your organism. System approaches fail. So this is where all the money is going in biology now. And so this is a bit like putting 300 in a carrier bag, crushing them, shaking it all up, and then taking the statistics of the final states of all these 300 Tamagotchi, and in the hope that it's going to tell you something about how a working Tamagotchi does it. It's not going to tell you much. So that's a challenging state that we're in biology. But the problem is it doesn't stop there. Because for biologists, they've got an even grander challenge, and it's dealing with the second of the three challenges I want to talk about today, which is loss of Earth's life support system. So everyone knows about climate change. Even the Americans have started to say that it exists, so there's a hope for us. But the problem at the moment is people tend to think of it as just as a carbon. If we stop producing carbon, we're all fine. And we don't really know what carbon we're producing, or how much carbon we'll produce in the future. But it's already having a big impact. It's not, and the reason why it's not just about carbon is because we're consuming the planet's resources faster than, well, not just faster, we're just consuming non-renewable resources, which is not a good idea. If we're having a big polluting effect on the planet, and we're destroying most of what resources we have. I just want to show you, so this is this Citarum River in Indonesia 2 decades ago, which isn't long ago, 20 years ago. It supported hundreds of thousands of people, both for food, water, jobs, and as you can see on the right-hand side, this is bits of the Amazon that are being chopped down for cattle and for soy. None of it is particularly pleasant. I just want to show you this. It's remarkable. This is a river, and of course, no fish live in this. It doesn't support anybody. People have to scavenge. One thing I will say about this: most of this is plastic. The UK uses 9 billion plastic bottles of water a year. The US produces and uses and throws away 50 billion plastic bottles every year. It takes a lot of energy to produce. Creates a lot of carbon, uses a lot of fossil fuels, uses 7 litres of water to produce one bottle that contains 1 litre of water that comes from Switzerland somewhere. It's not anyway. We're not living sustainably. I know I sound like some sort of welly-wearing Greenpeace demonstrator, but this isn't my conclusions. It's conclusions of the Millennium Ecosystem Assessment Group, which was all the great and the good who met for 10 years, produced a report in 2005, and it makes for depressing reading for all the reasons I'm just about to come to, including the one that I've just mentioned. One of the most important things, one of the most worrying things, is that the consequence of what we're doing, both with carbon production, increasing carbon production, and rising temperature already, as well as loss of habitat, use of gross misuse of resources, we're fundamentally changing the Earth system, which isn't just about carbon, in ways that we can't predict and don't understand, but it's a system that we rely upon for our survival, we and hundreds of thousands of other species. Why is it related to biology? Because 70% of all the world's CO2 is sequestrated by biology, by biotic aspects of the Earth system, forests particularly, but also microbial communities. It's the things that we're destroying habitat to 80% of the world's biodiversity. A remarkable 50% of the entire biomass of the planet is microbes. I'm going to come to that in a moment. Things that you can't see. There are absolutely trillions of them in this room. I'll give you one good example. 1 gram of soil contains 10 to the 9 microbes and about 10 to the 5 species. We are fundamentally changing that at an alarming rate, and it all forms the basis of the entire planet's bioenergetic network, the food webs that we ultimately rely on for our own food. The Millennium Ecosystem Assessment Panel quite rightly concluded that in geological time, we've embarked upon the greatest mass extinction of life on the planet, certainly far greater and far faster than the thing that wiped out about 80% of all life on Earth 65 million years ago, which is worrying. And as a consequence of that, we are beginning to witness rapid loss of, as I say, the life support system that we depend upon for our survival. We're already seeing food prices go through the roof, severe water shortages, wars over water beginning to look as though they're about to start breaking out. I mentioned pandemics earlier. We're creating absolutely wonderful conditions for a global pandemic to emerge from nowhere, which, as I say, if it kills 1.2 billion people, we're all in trouble. The other 5 of us that are here. There's no evidence whatsoever that the planet, even if we do something fundamentally change what we're doing, that the planet's capable of supporting supporting 9 billion people, which will be on the planet, as opposed to the 6 that are on the planet now in 40 years. So I just want to say something about the state of, we've gone through the state of biological science, so I just want to say something about the state of environmental science and why biologists are needed to help sort the problem out, or at least understand it. This is taken from the Hadley Centre. So in the 1990, climate models were just something about simple atmospheric physics and terrestrial physics, about oceans and clouds and temperature. The physicists who were running the show at this point realized that by 2001 that they needed to think about something about the carbon cycle, well done, and something about aerosols, and there was something that might be interesting about circulation and about feedback systems. It's still physical, and that produced a fairly alarming increase in poor outcomes for the planet in the IPCC predictions by 2001. 2007, this is really state-of-the-art climate modelling. We've got atmospheric chemistry. The chemists have got on board, as well as the physicists. They are beginning to realise that there's this thing on the planet called vegetation, which might have some impact. So they've got this model of in Earth system models at the moment on vegetation interaction, which is euphemistically called because there's not a lot of vegetation involved or interaction. And I'm going to come to that in a moment. But this really produces the IPCC predictions on which we're all relying for determining how much carbon will be in the atmosphere and what temperature consequently will be by the end of the century. And we're now past 2 degrees, which everyone thought that we might be able to limit, and up to 4 degrees, most IPCC scientists now saying, and that's before we actually really deal with this biological bit, vegetation interaction, and I'm going to come to why that's important. Even in this current state of the art of climate models with this vegetation interaction, we have no idea how to include the role of forest dynamics properly, or microbial communities. Forest dynamics are important for reasons I mentioned. Sorry about all this text. This comes from Steve Pacala, who's probably one of the world's leading climate scientists in Princeton. And he's a good friend, even though I've just pinched some postdocs of his. But anyway, it's another story. So I'll just read this out because it's important. Plant communities are vital to Earth system regulation, yes, but their role and sensitivity to change is currently barely understood. Scaling from physiology to ecosystems is currently skipped over in Earth system models, ignores non-linearities in whole plants, interactions between plants within species or interactions between species, how whole plants to respond in terms of growth, mortality to resources and competition, and changes in their environment, so climate change, and dynamics of the forest community. The yellow bit is important. This is the single most important outstanding issue in Earth system modelling. So everything that we're betting the farm on in terms of economic policy, energy policies, are woefully inadequate and carbon reduction policies because they don't take into account this, and it will have a fundamentally important contribution and difference in climate models. We don't know how to model role of microbial communities. I've taken one coccolithophores because they're very important. Not only are they the basis of all the planet's food chains, food webs, as I say, ultimately including the ones that we rely on. They also produce dimethyl sulfide, which is an important contributor to the formation of clouds. And if there's no clouds, then the planet's going to heat up even more quickly. They have all sorts of other carbon sequestration qualities. If anybody, by the way, anybody who thinks that coccolithophores is this esoteric sort of organism, they're 5 micrometers across, so they're pretty small. But in communities, they are the size of continents. I might have a picture somewhere that shows you a coccolithophore community. It's about the size of Europe. And anyone who is familiar with the White Cliffs of Dover, it is solid coccolithophores. It's not rock, which is interesting. So it gives you a sense of how at least how frequent they are, but that's changing quickly. And just to illustrate the point and hammer it home a little bit, in this 2007 model of this crude vegetation interaction, we can see the difference it has. So this is prediction of temperature change as a consequence of carbon in the atmosphere to the end of the century. The blue line is when the physicists ran the show. This is from NCAR in the US. And that predicted a rise at the end of the century of 2 degrees. And then once the chemists got involved, it was 3 degrees. Now the biologists are getting involved, even in this crude sense. Well, actually, it's the ecologists, so that tells you everything you want to know. We're up to 5 degrees, and there aren't many people around who would seriously say that the planet's going to be habitable for us as a species at 5 degrees. It's certainly not going to be comfortable to live on. Our own modelling work, in including this dynamic global vegetation modelling, which I'll briefly mention, which is doing the job properly, as Steve Pacala suggests you need, or at least starting to think about how to do the job properly of modelling forest dynamics, shows a 6-degree rise by the end of the century. And I'm pleased to say that our model has been taken in by the IPCC. That's good. That's good news. The bad news is it's going to be in the one after the one that's just been produced, so it's going to be nearly a decade before our model ends up in the IPCC report. The worrying thing is it will almost certainly predict a rise in temperature by the end of the century of 6 degrees, and there's no one seriously who works in the area believes that we can survive as a species at that temperature, and we're already here. So I don't have children, I don't have grandchildren, but for those that do have grandchildren, they are not going to thank us for the planet that they have to live on when we've all gone. And I do feel we all really ought to do something pretty urgently about it. So the state of environmental science and state of biological science is not a great one. Halfway through this talk, you'll be pleased to know I can draw some conclusions. The first is that what's pretty obvious, that we need to urgently better understand complex natural systems, very urgently, I think. Second conclusion is that traditional scientific approaches continue to fail. In all these, in the 2 fundamentally important societal challenges that we face, and I'll come to the third one. It's just the same. Some barriers, some fundamental barriers exist to further progress in science as a consequence of current scientific approaches, and we need to address those very urgently. And it's not clear to me to what extent e-science and e-research, what contribution it can make to that. I hope it's an important one. Conclusion 3, however, is that as we've seen, collecting petabytes of data is not the answer. We're currently doing that. We've done that in molecular biology, and the Human Genome Project doesn't tell us much about life. We're busy doing that with systems biology. It's probably not going to tell us much. The Large Hadron Collider has probably produced more petabytes of data in its day of working than most biology has done for the last 50 years. All to try and find at least evidence. They know they're not going to find it, but at least evidence of these particles that they've made up. And did I say that? Gosh. And it's recorded. Can we not release this? Anyone can give good answers for me of what a lepton is doing well. Anyway, that's a different talk. So conclusion 4 is, yes, I think transformation of science is urgently required. We need, for a start, we need to radically rethink biological science, in my view. Like I say, this is a personal view. It may come as no surprise to you that it's not one shared by many people, but I was asked to give a talk, so here it is. So claim 1 is, I think, where we might find some of the answers is to reformulate the problem. So actually ask some different questions. And there's a lovely quote from Einstein and Infeld from 1937, which is, the formulation of a problem is often more essential than its solution, which may be merely a matter of mathematical or experimental skill. To raise new questions, new possibilities, and the important one here is to regard old problems from a new angle requires creative imagination. I'll come back to that at the end of the talk, and marks real advance in science. First of all, I think there's a dreadful paucity of creative imagination in science and scientists, and that's, I think we need to do something about that. Claim 2, bear in mind, it actually exists in 10-year-old scientists, and I don't know what happens between the age of 10-year-old scientists and undergraduates, but something dreadful goes wrong. Claim 2 is that I think that some of the answers were found in radically new conceptual and technological tools. And claim 3 is, if we can do both, if claim 1 is true and claim 2 is true, claim 3 would be true, which is that it would form the foundations of a profound, I think, new scientific revolution. That is indeed a bold claim, because scientific revolutions are indeed rare, and indeed, there's a case been made, there's only ever been one. It was the scientific revolution that occurred in the 17th century. And when it did occur, it was underpinned by new conceptual tools, new technological tools, and people with a different way of thinking. And I just want to illustrate that point by what should have been the title of this talk, which was the codification of heaven. So, up until the late 16th century, there was this geocentric view of the world, which everyone was all for. And along came Copernicus rather than Galileo. Everyone thinks that all this was driven by Galileo, which is obviously completely untrue. And then reformulated the problem of some inconsistencies with this with this geocentric view of the heavens. So in 1543, Copernicus produced this De Revolutionibus, which was interesting because it introduced a new conceptual tool into what was then natural philosophy, which was codification. So this is, by codification, I mean a precise, formal, testable account or explanation or description that enables a precise hypothesis to be formulated and tested using algebra, which is interesting. So something that was a bit late to come to Europe. The Iranians had been using it for 3,000 years, but better late than never. New computational tools. So after 1543, along came Kepler, who in 1596 developed an actual model, a computational model with predictions of the solar system. So this was the first ever prediction machine. And I'll come back to prediction machines because it's important about models and the role of models. And then finally, Galileo came along and actually and used a new technological tool, which was a telescope, to actually not discover that all this was happening, because it was already known by Copernicus 50 years earlier, which was to test parameterize what we would now call test parameterize and constrain the model. The reason why we're all familiar with Galileo is because he got himself into a bit of deep water because it was to do with sunspots. And at that point, the church considered heavenly bodies, including the sun, most importantly, to be perfect. All heavenly bodies had to be perfect. And loads of people had seen sunspots before. But what Galileo did was see what everyone else had seen, but thought what no one else had thought, which was that these weren't something that were going in front of the sun, they were on the sun. And so they had these imperfections, these spots on the sun, and that got him into deep trouble with the church because you couldn't have imperfect heavenly bodies, and so that's why Galileo is better known than Copernicus. Yes, he got into a bit of bother. Anyway, so new technological tools, telescope enabled this. After that, Newton did the codification of Earth using another conceptual tool, calculus. Again, he'd stolen from the Iranians, really. So he published Principia Mathematica in 1687. So the other thing that they all had in common is creative imagination. They were able to reformulate some pretty thorny, intractable current problems of the day. What I'm arguing is that what we need to do next, after codification of heaven and codification of earth, is codification of nature. So the new kinds of conceptual tools that I think are at least a good start are things that come from computer science. I'm not a computer scientist, I'm a neuroscientist, but one of the things that I decided to join Microsoft for, to sort of do all of this stuff, is that I am convinced that computer science has some of the computer science rather than computing has some of the tools that we need to make a start. So these are things like treating complex natural systems as abstract state machines. So thinking about levels of abstraction in science are going to be important to start to tackle this problem. Things to do with processes I mentioned are important in complex natural systems, and process-based algebras also seem a good place to start. Things like automata, finite state automata, cellular automata, again, interesting conceptual frameworks for thinking about interaction between complex natural systems, as well as traditional mathematical approaches. I'm not sure, I don't see the point in throwing the baby out with the bathwater. The reason why I think I would want to summarize why these tools emerge from computer science, theoretical tools from computer science, is that computer scientists tend to think about how the tools for modeling, building, and predicting the behavior of highly complex engineered system that's characterized software, that's characterized by time dependency, distributed control, is highly dynamic, is highly concurrent, is highly reactive, and they're all the terms that biologists use to talk about living things. So the theoretical tools for studying the world's most complex, dynamic, reactive, time-dependent, concurrent engineered systems, software, and you could be forgiven for thinking we don't use these tools in Microsoft, but we do, are some of the very similar tools to the kinds of tools that biologists need to study highly complex, dynamic, reactive, time-dependent, concurrent natural systems, living things. And it's proving very fruitful in my lab at the moment, and I'll maybe get some chance to give a couple of examples. New kinds of technological tools. Also, we've started from tools from computer science. If you want to model, build, and understand highly complex, all the terms I use there, predictive, dynamic, reactive, complex systems and processes, what computer scientists do in building them is build a programming language. And so some of the technological tools that we're looking at is building programming languages for modeling biology, and complex biological processes at multi-scale levels of modeling and of systems. This is a model of a pancreas development, by the way, which I'll come to briefly later. Again, I think this is the initial work in our lab, and we're not the only lab doing this, but I think we're probably, sadly, the lab sort of pioneering this work, and there should be a lot more groups doing this stuff. But it's early days, but it looks very promising. The combination of these new kinds of conceptual tools and new kinds of technological tools will hopefully enable us to develop new kinds of prediction machines. So not about simple physical things like heaven, but highly complex natural things like ecosystems and how complex natural systems and living things interact with each other. If we can do that, and it is early days for us, and I'm making a claim here that this is a good way to start. I'm not making a claim that we have all the answers. It would completely revolutionize our understanding of how living things work, but from biochemistry to the biosphere. As I say, in my view, they're the most urgent problems we need to tackle for our society this century. I do have time, I think, to give you some very brief examples from my lab. I'm not going to go through all of them, so my apologies for the slides I skip. The first one I mentioned is the codification of life. So instead of using just ordered differential equations or reaction graphs, we've taken some mathematical, i.e., so a formal, precise account or way of representing complex biological processes. In this case, this is actually a key function in the immune system called antigen processing. So there's a mathematical representation. We've then coded that up so it's into this precise formal programming language that we can then model all these, all the interactions, processes that occur in this immune system function or any biological process. And I use this one is just an example, but we've got it for many, many examples of biological processes. And capturing the dynamic aspects and complex dynamic predictive modeling is unheard of in biology, but we've got using this approach, we've got some examples of the first, I think, really compelling examples of predictive biology. In addition to that, we've done some things to make this programming language biologist friendly. So instead of having to type millions of lines of code, you can do things like drag and drop proteins and way in which they interact together just onto this canvas, link them up. They've all been sort of biologically proved correct, and then you can actually read the output either as this kind of interaction that experimental biologists would understand, or in this visual description of how maybe theoretical biologists might understand. Examples of using this is, I'll just mention this one because it's an important one, because the immune system is the one thing between us all and the cemetery, and we don't understand how it works. And so the current kind of paradigmatic view of doing immunology is either using something like scanning electron microscopy to, this actually is a T cell that's been produced to attack cancer cell. The purple thing is a T cell and the green thing is a cancer cell. Biologists will either use scanning electron microscopy to show that how these T cells physically attach themselves to a cancer cell and make some inference about what processes are involved, and then they will produce a model of that, and it literally is a cartoon. And what they will say is, for example, tapacin, which is this, is involved in antigen presentation, which is this whole complex, and this is actually a cartoon. This is state-of-the-art model of how immunologists describe and try and explain a model, in this case, class 1 MHC, class 1 major histocompatibility complex antigen function, which produces T cells, or leads to the production of T cells to attack either a virus and stop attacking the body itself when there's no need. What we've done is use this programming language for biology, which is actually called stochastic pi machine, because it's based upon stochastic pi calculus, which is a process-based algebra, to develop an entirely predictive model of the world's first ever entirely predictive model of MHC class 1 function and T cell production using the approach. This is all very familiar to immunologists. The model makes lots of predictions about role of tapacin, antigen presentation, T cell production over time. All these predictions have been borne out experimentally, and there's a paper being written up for Nature Immunology as I speak. But this is important stuff, because lack of quantitative predictive models of immune system function is the one thing that's preventing fundamental advances in a really promising area of medicine called immunotherapeutics, which is to get, largely crudely put, is you get the immune system to solve many problems that we rely on drugs on at the moment. So this is the work of Andrew Phillips, a scientist in my lab working with two immunologists at Southampton, Jörn Werner and Tim Elliott, and some colleagues at Weizmann Institute. Briefly, understanding the development of life. Organogenesis is one of the key functions for producing things like us or mice, and it's the process by which cell proliferation and differentiation occurs from stem cells and to produce lungs, heart, you name it, crudely put. Again, we've been using this programming language to build the world's first ever entirely predictive
model of organogenesis. This is actually a pancreas, but we've got some other organs. And so we use this programming language in which the basic level of abstraction is a cell, and it's this executable component of it which is important. We then link that to this kind of realistic-ish 4D, including time, output of this model to show that the developmental process of a pancreas. Link that to the output of the model is also amenable for mathematical analysis of organogenesis. This shows cell production and then differentiation and proliferation. Interestingly, because anyone could build that sort of model, is that it actually makes all sorts of predictions which have been borne out in numerous parts, numerous experimental approaches and one thing or another. So the first is the model conforms to real developmental processes and histology, so the way in which the model works, and bear in mind there's nothing in this model in which we code it up that actually determines that it should become this shape, the shape of a spleen, the shape of a pancreas. It's actually an emergent property of the model, as it is an emergent property of the system. Being able to account for emergent properties in biological systems is unheard of. So this model is mildly interesting. The model conforms to real development of pancreas and from histology. More interesting is the emergent properties of the system. This is the model. This is actually microscopy images. It's explained by the model, and I won't, in the interest of time, I'll go into details. But most importantly of all is that you can do in silico, i.e., in a computer, realistic experiments on this virtual pancreas development and the process of organogenesis generally, and all of those in silico experiments we've actually done, or our colleagues in Weizmann have done, in vivo on real pancreatic development, and the model predictions are absolutely borne out almost fully by the in vivo experiments, which is again unheard of. This is work done by Yaki Sethi, a postdoc, unbelievably, in my group, together with some colleagues at Weizmann, David Harrell, and some others, and Hillel Kugler, who's also in my lab. Very briefly, I just want the last one. Just want to talk about next generation Earth system science. I read out that quote earlier from Steve Picala about importance of. Do you want to get that? About the importance of forest dynamics. Has he left it in his bag? Yeah. He's gone out. Maybe it's just out of embarrassment. I'm quite keen to know who it is now. Tell us when you know. Anyway. So just very briefly, so all this forest dynamics stuff, we've started to try and tackle all that forest dynamics stuff that Steve Picala mentioned. It's a non-trivial task, but we've built this model, this dynamic global vegetation model, which is everything from tree xylem and respiration of a single plant up to forest stands, which are about one hectare wide, about 1,000 trees, up to entire forests. We've constrained the model with lots of real-world data. Was it important?
It's apparently 9 a.m. on the East Coast.
It is, yes.
In case anyone was curious.
No, not at all. Anyway, and done all this sort of interesting stuff, which is all this nice visualization to enhance creative imagination in exploring these models. This is the model that's gone into the IPCC. It's driven by, it's all been done by Drew Purvis, an amazing chap that I'm lucky to have in my lab. Completely bonkers, but it's nice. But it's nice. It's actually nice to be able to attract. It's not the subject of this talk, but there's something quite satisfying about being able to attract British scientists away from Princeton back to the UK. And we've got a few of those. That's a different talk about the state of education in this country. Anyway, so that's
just to conclude, that's all. I suppose the general point I want to make is the kind of approaches that I mentioned earlier about these new conceptual tools, new technological tools, and a different way of thinking by people like Drew. I'm too old and too stupid to be of that ilk, but I at least have some clever people in the lab, can make an enormous difference. I'm not going to use that one because I haven't got time. We're clearly starting to do something right because we've only existed 2 years, and already we have publications on the work that I gave you 3 examples out of about 40 projects, but we're already producing publications in PNAS, in Nature Biotechnology. We had 2 papers in the same issue of Science, Proceedings of the Royal Society. I know it sounds a bit boasting to say that. I'm just saying that by completely rethinking approaches to science, it can make it looks promising, can make a big difference. So just to finish off, because I realise my 40 minutes is up, if we can tackle some of these, well, to sum up, we urgently need to solve some of these major challenges for society, because we're in trouble if we don't.